we are trying to push forward precision oncology for breast cancer. And this is probably something that's a little bit new. Some may say this is more science fiction than it is fact, but hopefully this will become more of a standard of care as we move forward in the decade. These are my financial disclosures. The overview of my talk today is really going to be a little bit of background on cancer, and obviously this group or breast cancer, I should say, and this group I don't really need to go over that extensively, but I do want to talk about what we define as precision oncology, because there are many uses of that term, and how we're actually looking at the DNA of both the patient, which we call germline, versus the tumor, which is somatic, and how we determine clinical actionability. A big component of that determination is through molecular tumor boards. And something that I will share with you are virtual molecular tumor boards, which came out of our interactions through the Global Cancer Institute, of which Bangladesh has representation, and then some future directions and conclusions. Again, in our country, this is, again, not new to anyone in this audience, but in our country, we have about 250,000 new cases diagnosed each year of breast cancer. And that ultimately translates into about 40,000 deaths each year in our country. So that shows you if we can catch breast cancer early, the majority of patients will in fact be cured of their disease. Yet because of the incidence of breast cancer, that absolute number of 40,000 is still a pretty staggering number. It's one of the leading causes of deaths in women in our country. And when we see patients, we often talk about early stage, which is curable, requiring both local and systemic components in many cases, and then metastatic disease, which we usually think is not curable. Although I would argue there are probably some metastatic breast cancer patients, particularly with HER2 positive disease, that have achieved cure despite having a diagnosis of metastases. Regardless, all metastatic disease nowadays can have some form of treatment, as we know, and we were just hearing about. I do want to take a little bit of time on this slide because we often get, as oncologists, asked the question, well, why is metastatic disease curable or incurable, but early stage disease is? What is the purpose of systemic therapies as a medical oncologist? And the way I like to think about this is that cancer is really a disease of DNA gone bad, and it has that problem, but the real heart of the problem is the fact that it has infidelity, leading to tumor heterogeneity. We know that the repair mechanisms of DNA in cancer cells is usually on and usurped for growing purposes, if you will, such that when one cancer cell divides, you get subclones, and those subclones get more subclones. And when we put a drug in treatment, by the time we can clinically see metastasis, there will already be a population of drug-resistant clones. When that occurs, there's a bottleneck, and then these cells start taking off and dividing, and again, they get progeny with even more heterogeneity and more pre-existing drug resistance population. The time we can really cure metastatic disease is ironically when we can't see it, and that's the so-called micrometastatic disease, which we do in the adjuvant setting. It's hard to kill patients, that's akin to here, but we haven't really developed enough drug-resistant clones so that we can throw 3 or 4 or more drugs at a patient's microscopic disease burden and eradicate that. And so to put, again, some numbers to this metastatic disease, minimally a cubic centimeter of cancer is about 1 billion cancer cells. And so we're talking really huge numbers of cells, and therefore pre-existing mutations are already present. So the goals of systemic therapies, as just outlined, in metastatic disease, we try to prolong life and treat symptoms. In the adjuvant systemic therapy paradigm, we're trying to eliminate micrometastatic disease. In our country, this translates to about 150,000 patients who are candidates for these additional systemic therapies in the adjuvant setting. Now, again, I already mentioned metastatic disease is not curable largely, but I also mentioned that we have a lot of drugs now in our armamentarium. And I think many in this audience already know this, but hormonal responsive disease, estrogen receptor, progesterone receptor positive disease, we can use single agent endocrine therapies. We have combinations now with CDK4/6 inhibitors, exemestane and everolimus is a combination, everolimus being an mTOR inhibitor, and certainly more coming down the clinical pipeline. In metastatic disease, we heard about HER2 positive disease, and again, there's really a huge number that are now available in our country, including antibodies as well as small molecule inhibitors. These can often be given with or without chemotherapies and hormone therapies. think I heard mentioned neratinib and tucatinib, which are also newer oral anti-HER2 agents. We also now have available for triple-negative breast cancer some options beyond chemotherapy. And so sacituzumab is a antibody conjugate that is against the COP2 protein, which is also now approved in this country for triple-negative breast cancer in the metastatic setting, as is an immunotherapy. Suffice it to say, choices must be individualized depending on the situation of patient's disease, including the receptor status. But I will take off from there and argue that really precision oncology almost began with breast cancer decades ago with the discovery of HER2, which is a receptor tyrosine kinase, and the realization that HER2 positive breast cancers were what we now call oncogene addicted to that receptor tyrosine kinase and that pathway, which is outlined here, the PI3 kinase pathway and the AKT pathway, which are downstream of activating HER2. So we have, as I mentioned in the prior slide, a number of therapies that attack this and are highly effective. And again, I would argue in some cases for metastatic disease, even cures. I think that this is one of the, I like to say, poster children for targeted therapies and bench to bedside type of research, because HER2 positive breast cancer, and as many of you know, 3 decades ago was in fact a worse subtype of breast cancer that happened in the house because that's possibly if a patient receives HER2-directed therapy. And with that, that's kind of how we view and I view precision oncology, is that we don't necessarily use burn and pass paradigm to treat all breast cancers the same. This is so largely true for early stage disease because this is curative intent, as I mentioned earlier. And so we don't really have ethically and medically a platform to really deny standard of care for early stage curative intent. So most of what we're doing in breast cancer for precision oncology nowadays is in fact in the metastatic setting. The rationale again is that we know cancers are unique as the individual patients who are afflicted by these diseases. And if cancer is a disease of DNA and we all have slightly different DNA or sometimes vastly different DNA, it would make sense that we should be able to understand the genetic underpinnings of a patient's cancer and be able to treat this effectively. How do we do this? I think many in this audience are probably familiar with next-gen sequencing, or at least have heard of it. Technically, this is kind of laid out here in this cartoon or diagram. Next-gen sequencing is actually very simple in terms of that we just take DNA, we chop it up into small little fragments, and we sequence both ends and overlay it across a reference human genome. The much more tricky parts about this is there are many different reference human genomes, and so there's a consensus. That makes it sometimes difficult because, as I said earlier, everyone's DNA is a little bit different. But needless to say, if we do this, and the key here is that we can sequence all of these little fragments, what we say massively in parallel, we can find unique differences in one strand versus the other. Here's an example of a single nucleotide polymorphism where 50% of the time you should have one allele and 50% of the time you should have another allele. And that's part out here. In this particular, here, and in this particular case, this individual has 50% of the A allele and 50% of the G allele. That's in contrast to this situation on this particular area of DNA where this patient is 100% homozygous for this particular T allele, and the reference genome says most people have an A allele. And that's really it, just distilled down to its basics. There's not magic. I know sometimes it sounds like this, but really it's just a matter of capturing DNA and then sequencing it many, many times so that we can get the highest level of sensitivity and accuracy. So as I mentioned, next-gen sequencing, or NGS, can sequence billions of base pairs of DNA in a time and cost-efficient manner. This does require, as you saw in the prior diagram, what we call redundant coverage. The human genome, the haploid human genome, is 3 billion base pairs, and if you were to sequence exactly 3 billion base pairs, you wouldn't have the whole genome, because those fragments of DNA will sometimes be overlapping, and so you'll have an area of the DNA, of the human DNA or genome, that will be sequenced 10 times, and other areas you will sequence none. And so to make sure that we sequence everything, we have to do this nowadays at least 100-fold coverage, meaning that we do 3 times 10 to the 10th, or 30 billion base pairs if you're doing the whole human genome. If we do this enough redundant sequencing, we can actually detect rare mutant DNA populations, down to as low as 1%. The other nice thing about next-gen sequencing is the flexibility to capture regions of interest. So for tumor sequencing, we don't do 20,000 genes, which is the human genome right now, commercially and institutionally. We usually have a panel of cancer genes. For those of you who know from Foundation Medicine and other vendors, probably a few hundred genes are sequenced. And the same is true for genetic testing for germline variants, meaning BRCA1, BRCA2, and other types of heritable cancer mutations. Typically, we only sequence now about 29 or 30 of those genes, even though, again, we have 20,000 genes in our genome. One of the things that I like to point out, though, is that nowadays that there's convergence of heritable or germline DNA sequencing as well as tumor or somatic sequencing. Why is this important? I think many in this audience already know and appreciate how tumor suppressor genes like BRCA1 and BRCA2 can have a huge influence in the incidence of developing breast cancer, or the likelihood. And that's kind of laid out in this schematic, where this is a pedigree of retinoblastoma, hallmarked here by the RB. So in pedigrees, circles are women and squares are men. And so this particular individual has an RB mutation in the germline, or what we call constitutional heterozygous. And in order for RB is also a tumor suppressor gene, just like BRCA1 and BRCA2. In order to initiate cancer, the second copy has to somehow be lost or inactivated. And there are a number of ways shown here that can occur. On the sporadic side, you can get retinoblastoma. It's much more uncommon if you don't inherit the RB gene. But when that happens, we say it's all somatic, meaning that the first genetic hit, as it's sometimes called, occurs just in the cell that's destined to become a cancer. But the mechanism of the second loss is identical to whether you have a sporadic or inherited form of retinoblastoma. And what is not really shown in that diagram is, in fact, there are multiple hits or genetic damage that are required for a normal cell, shown here in green, to go all the way to a cancer cell, shown here in red. And with that has to have a concept of clonal expansion, which is something Peter Knoll put forth in the 1970s at the University of Pennsylvania. What this really means is it's a mathematics numbers game. If you had the same genetic hit happen in just one cell, we'd probably never get cancer, because statistically that would be unlikely. Instead, what really is put forth is that after the first hit or genetic damage, you have to increase the population here so that there's some chance that a second genetic hit can occur. And again, more clonal expansion so that statistically this group of cells now that has two genetic hits, one of them can get a third one, and so forth and so on, until cancer ensues. So that's really important concept, because we want to think about for both diagnostics and for targeting therapeutics. We want to think about that because we want to usually go back, and if we can find anything in the patient's tumor that occurs early, you'll have to remember that this genetic damage is actually going to be propagated in all the progeny, whereas this one will only be in a subclone population. So the germline testing or genetic testing classic examples include what I had already mentioned, and this audience I'm sure is very familiar with, including BRCA1 and 2. That testing has evolved over the time. Before next-gen sequencing, it was a really difficult, laborious, and expensive task to perform these types of tests. Now with we've replaced old standard. And we can do, as I mentioned earlier, panel gene testing, which means that instead of just doing the BRCA1 and 2 genes, we can add other genes known to increase breast cancer susceptibility, such as TP2, TP3, and so forth. It's still typically a very small number of genes, as I mentioned, relative to whole human exome, as we call it, which is 20,000 genes. Now, the reason why there's been convergence of germline testing with tumor testing is, as many people know, we can now do something about this more than screening blood relatives, but it has therapeutic action. Many of you know that PARP inhibitors now are approved for BRCA1 or 2 germline gene mutation. And in this country, at least, we also have anti-PD-1 or immunotherapies that are approved if patients have a hereditary syndrome called Lynch syndrome, which is a germline mutation in one of the mismatch repair genes. And there are likely to be others that are approved that certainly scientifically make sense from an actionability standpoint today. Tumor-only testing used to be, and still is in many places, the standard. But in the past, this involved only a few mutations, what we call hotspot mutations. Then it evolved into cancer gene panels with several hundred genes. There probably is a point in time where I see full exome sequencing, so doing all the 20,000 genes will become more standard as technology improves. The harder part about this, though, is we've gone from a few hotspot mutations to several hundred genes, and certainly with whole exome sequencing, distinguishing what are really tumor alterations and whether they're actually actionable become very, very difficult. And so this has kind of turned our thinking about how do we use those data and information from the tumor only to actually afford the best care for our patients. The other thing that's distinct about tumor-only testing from germline testing is that generally no consent is required from the patient. So if one were just to do the tumor, then there's no consent, as opposed to genetic or germline testing, where in this country, at least, we have to make sure that there's pre-test counseling and post-test counseling, as well as a consent to get those genetic tests. Although there is, I think in some cases, great utility for some of these mutations, there's no definitive proof that testing all cancers affords general benefit, and that mostly this is performed in metastatic disease, at least in this country. I think that's an important point, and there have been some studies that show yes and a little benefit, and some studies that show none. I think those studies in the past have largely been flawed because they kind of put all cancer types and patients together. I think everyone in this audience recognizes that comparing breast cancer to small cell lung cancer is very, very different. So the outcomes, I think, have to be interpreted with a bit of caution. Now, as I was mentioning, it's becoming increasingly difficult to know what is in the tumor by doing tumor-only sequencing versus what is actually heritable. And I will show you in the next few slides some examples of why that can be important. But along the way, testing has now evolved at many institutions and companies to sequence both a matched normal germline DNA sample along with the tumor tissue so that one can filter out electronically from the computer what we call germline variants. And that way you can see, quote unquote, only tumor mutations. The disadvantage of this approach is that it's all done on the computer. There aren't human eyes looking at what's actually in the germline. And so when the data come out, one may never even know or see the fact that that patient could have had a heritable real germline mutation such as BRCA1 or 2, because that would have been filtered out by the computer algorithms. So that's missed opportunities. And so there are ways that companies are now getting around that. Some are reporting those variants and saying that these are in fact germline, not just in the tumor. That's a little bit challenging sometimes because, again, that does require consent. Other companies will just not filter out select genes, meaning that if you get a BRCA1 or 2 result back, the germline mutations will show up. They're actually telling you that they don't call them germline or not. They just call them tumor-only sequencing. The other challenges that come about from this are that one has to be very, very careful when getting these test results and knowing what was used as a source of tumor and what was used as a source of normal DNA. This is an example when I was at Johns Hopkins, that the tumor was a salivary gland tumor, or right people's buccal mucosa, but the source of normal DNA was saliva. And probably many of you in the audience right now are thinking exactly what we thought. This is not a good source of normal DNA because it's going to have contaminated tumor cells due to the location of the cancer. This is another one, which I'll show the result in the next slide, where there was a prostate cancer, and it was low pathologic tumor purity, meaning that it was surrounded by a lot of normal cells. But the source of normal DNA was not provided, and so companies generally will still do tumor-only sequencing, even if they provide germline filtering. If they do not receive a normal paired sample, they'll go ahead and just do the tumor. Why that's important is that if you get a result, you can't tell, again, whether or not this is just in the tumor or not. In this particular case, an individual colleague of mine found what he thought was an androgen receptor mutation in the sample, but it turned out it was in fact just a normal variant of the androgen receptor gene in that particular patient. It becomes even more complicated with what we now call liquid biopsies using cell-free DNA. This is an area of research interest of mine and now commercially available at many countries around the world. Circulating cell-free DNA or CCF DNA, often called cfDNA, refers to these small DNA fragments that are shed by all cells into the circulation, so both tumor and normal cells. Circulating tumor DNA, ctDNA, refers to the DNA shed specifically by tumor cells. And then we like the term plasma tumor DNA sometimes because in the old days, people were using serum, plasma, lots of different analytes to actually look at circulating tumor DNA. But this specific moniker plasma denotes where the actual ctDNA has come from. This is shown schematically in this cartoon. The cancer cells here are blue, and you can see that their DNA or free-floating cell-free DNA is shed into the circulation, and that's here, denoted here by the different colored DNA fragments representing tumor mutations and alterations. Healthy tissue is shown here in the green, and it's a uniform normal DNA. We can collect this, separate it, and then use this for next-gen sequencing just as we would DNA from a tumor tissue. What's not depicted in this cartoon is the relative proportion of what we usually find, and that is tumor DNA is, cell-free DNA, I should say, is very, very small percentage-wise compared to the amount of normal DNA, which obviously makes sense because we have trillions of normal cells and maybe billions of tumor DNA. So it's usually a very, very small percentage. And that becomes almost like looking for the proverbial needle in a haystack when we're trying to isolate and sequence and find ctDNA from cell-free DNA. Most tests display what we call the allelic frequencies, which I mentioned before, and that's how much of that percentage of cancer DNA is in that sample. Germline variants, meaning heritable DNA, is fairly easy to detect because, as you can imagine, if this is heritable DNA, usually BRCA1 and 2 mutations, as an example, are heterozygous. So 50% of all the cells will have a normal copy of BRCA1 and a germline mutation. And so plasma will reflect that because it reflects, again, both normal and cancer cells. Clonal hematopoiesis is also something that is relatively new, but it turns out with cell-free DNA, you can sometimes detect patients who have the earliest changes and mutations of hematopoietic malignancies. So this is often called clonal hematopoiesis of indeterminate potential or CHIP. These are now usually easy to spot because we know what genes they are, things like JAK2, sometimes TP53, and others. Again, these are the same genes that are mutated as in myelodysplastic syndromes and acute leukemias. And so we now understand that these may not actually have any consequence. In fact, it seems like the biggest consequence is that it predisposes to heart attacks. But they can also be the precursors of hematopoietic malignancies. Because of all of these variables, there's often much confusion. And so this is a case reported of a 73-year-old with metastatic breast cancer, was originally diagnosed in the 1990s, had a strong family history of breast cancer. At the time that she was seen at Johns Hopkins, we never received germline testing. There was not an easily biopsyable lesion. And so we sent off a liquid biopsy with cell-free DNA. I apologize, you can't read the top here, but you can see the genes. PIK3CA, these are what we call hotspot mutations in an oncogene that is very commonly mutated in estrogen receptor-positive breast cancer. And here again, looking at the frequencies, this is only 4 to 4.5% of the total amount of cell-free DNA. These are mTOR inhibitors, which again, companies will put on sometimes what they would recommend. I would caution everyone to look at this with a grain of salt, if not sometimes I could say a block of salt, because many times these are based on literature that is not really at the highest caliber or highest definitive conclusive calculations. So that's really where one has to be careful. And so this patient also had in the blood a COP1 mutation. Now, if you look here, this is even smaller percentage than this. So it's very unlikely, and then also some TP53 mutations. It was very unlikely that this patient, in fact, had a germline, because as I was mentioning earlier, if this patient had a germline BRCA1 variant, this should be 50%. So this really spoke to us that this was probably a subclone population derived from the patient's cancer. And what to do with this? Well, I'm going to mention this in the next few slides, but I convened when I was at Johns Hopkins a number of years ago, one of the first molecular tumor boards, and we met to discuss this case. And we felt like that this was a truncating or really loss of function BRCA1 mutation. And given the family history, it was still plausible that it was very plausible that this was a somatic mutation, though probably, as I said earlier, in the subclonal population. We still recommended germline testing. It's something that we would do because we remind everyone that these tumor-only testing are not substitutes for germline testing. And that was actually negative for any evidence. So we felt like because of the subclonal population, it was probably unlikely that a PARP inhibitor would have benefit over a PI3 kinase inhibitor trial that was actually at our institution. And so that was the recommendation of our tumor board. I think that goes to what I'm getting at is for precision oncology, the need to have some sort of content expertise or tumor board that we can discuss these cases and pull together, just like we do for any other types of tumor board. Group experts can ask a variety of questions. I'm not going to go into all these questions and show you the publication which you can look up that describes this in detail. But I think suffice it to say, the challenges of figuring out what is real or what is truly actionable and what is not an easy task. And so my argument is that molecular tumor boards are really needed if one's going to do a lot of NGS on these tumor specimens and liquid biopsies. So this was the first tumor board that I started called the Genetic Alterations in Tumors with Actionable Yields, or Gateway. And again, this has been published now for a number of years in JCO Precision Oncology, describing the experience of our molecular tumor board when I was at Johns Hopkins a number of years ago. We pulled together a lot of different expertise, just like a normal tumor board. This would include, however, a slightly different makeup. We had medical oncologists from diverse specializations, molecular pathologists, importantly, especially now, as I was mentioning about germline testing, genetic counseling. We had phase 1 clinical trialists, patient advocates, many fellows and residents and students, research coordinators, and even legal expertise. That's important because we're trying to anticipate or expect the unexpected. The University of Michigan had a case where a man with prostate cancer, as an example, they found with next-gen sequencing proviral HIV DNA. And the patient wasn't aware of this, physicians weren't aware of this, and that presents a number, again, of ethical and legal dilemmas that need to be vetted. Sorry. So our tumor board locally at Hopkins made written recommendations for our providers regarding the actionability of alterations. We like to think kind of in this order. The first thing that we'd like to recommend is eligibility for FDA-approved drugs in that tumor type. And then also second to that is FDA-approved drugs for the same alteration in another tumor type or so-called off-label use. And then it goes down with lesser levels of evidence. When I arrived at Vanderbilt after really 20 years at Johns Hopkins in 2018, one of the first things I did was establish our own molecular tumor board here in the Vanderbilt Ingram Cancer Center. And this is called the Hereditary Oncology Personalized Evaluation Molecular Tumor Board. And the hereditary part is very key and purposeful, as I was mentioning earlier. We really do germline testing in parallel with our patients who have cancer, and our genetics are intimately involved with all of our tumor boards. So we meet weekly, discuss germline and tumor NGS results, and make recommendations for the providers. Now, what has happened along the way is this has grown, and it really started the Global Cancer Institute, which I think all of you in Bangladesh are familiar with. This was started by my colleague at Massachusetts General Hospital. The genesis of this really came from the Avon Centers of Excellence at Johns Hopkins. I was the site PI, if you will, for that particular mechanism, and Dr. Doss was the site PI for MGH. And he really also created or helped create the Global Scholars Program. This brings together, as I think many of you know, breast physicians, surgeons, radiation oncologists, et cetera, all over our country and all over the world. It's a nonprofit organization and meets monthly and has been using Zoom well before the pandemic. I think Paul was a real visionary in that regard, that we could really bring the world closer together using the internet. GCI, and this is the power of connecting people. And these are all the sites. I'm sure it's grown by now because this is probably a dated slide. But these are all the US and global sites that are involved. And why this is important in terms of the story and how it evolved my thinking for molecular tumor boards is because of a case from Bangladesh. And I think some in the audience may even know the story because you may have been involved in this girl's care. But there was a case that was presented in the summer of 2016 of a young girl in Bangladesh who was presented to the GCI breast tumor board. And one of the things I like to say is that I'm the only geeky physician scientist who runs a lab in that tumor board. And this was called Juvenile Secretory breast carcinoma, which many people had not heard of. In fact, I think most people have not heard of. And it was interesting. It's almost fate because I happened to be on that tumor board that one fateful Monday. And it was presented. These tumors are usually cured with surgery alone, but she had, unfortunately, 3 or 4 recurrences and widespread metastatic disease. This cancer is much more common than a classic breast tumor, and it is not responsive to chemo or radiation therapies. But I also happened to be at the time on the scientific advisory board of a small startup company called Oncology, and they had a drug which was really an incredible drug in terms of its specificity. And they were looking for patients because there are only a few cancers in really the whole cancer spectrum that have what we call NTRK translocations. There are 3 NTRK genes, 1, 2, and 3. The drug targets any of them, but the real predictive marker is that there has to be some sort of translocation. And so I also happen to know in my kind of geeky physician scientist role that 20 plus years earlier, there was a paper in Cancer Cell describing that all of these juvenile secretory breast carcinomas, with very few exceptions, had a translocation of ETV6 and NTRK3. So I got the patient's tumor over to Hopkins, and then I forwarded it because to NSK, Memorial Sloan Kettering, where the trial was being held. And they performed staining and next-gen sequencing and verified the actual alteration. Now, this is the girl when she got to MSK. You can see this is a left tumor mass, and this is, again, I think, the 3rd or 4th tumor recurrence. And she gets on this study of the drug, larotrectinib, and within 2 months, she has complete resolution of her disease. The CAT scan also is equally dramatic. The mass here is gone by day 60, and pretty much all of her lung metastases are also gone by day 60. And I should mention Dr. Baki, who was a physician over at Bangladesh, is a part of this collaborative study. Since learned that she did have a local recurrence again, and they were just small nodules that were biopsied, and the company had already had in place a 2nd-generation inhibitor. They had anticipated a resistance mutation arising based upon preclinical data, and she went back to the Memorial Sloan Kettering on that trial with a 2nd-generation inhibitor, and also discussed at GCI a couple of years later, had another complete response. And to my knowledge, because she was presented again recently, she is still doing okay. And so she's taking the pill. This is one of those areas where we don't really have data to guide us how long she should be on the pill and what her overall prognosis is. But I believe the decision was made that she should try to stay on this for as long as she can. That really inspired me that we could really be helping a lot of people more than just through the breast cancer or GCI breast tumor board, but maybe even through a virtual molecular tumor board. And so a few months later, after starting up here at Vanderbilt, I created a phenocopy, if you will, of Paul's GCI breast tumor board called Our Cancer Genomes. This is an international virtual molecular tumor board. We meet monthly, just like the GCI tumor board. Today, we have about 17, maybe more than that now, U.S. academic sites, a number of community practices in our country, as well as 6, I think maybe even 7 international sites. So I'm well familiar with the challenges of getting a time that's mutually agreeable for all different countries in the world. How this works is that we have a Redcap instance, so this is a secure data upload. There's also this scanner code that one can actually get to that Redcap database. We meet the fourth Monday of every month at 9 a.m. Central Time. We have a HIPAA-compliant version of Zoom. We redact everything, making sure that we're trying to use best practices to preserve patient anonymity. And then again, if people in this audience have any interest in this, please be sure to contact me and I'll get to you as soon as you can. This is what the Redcap database looks like. So you scan that barcode or go to the actual online, and this is what pops up. We try to make it as simple as possible. And then this is something that we provide for our local tumor board. We're not, for compliance reasons, putting out reports for international, but we do have the capacity to record them just as we do for the GCI-based tumor board. This is some of the group that actually convenes and discusses both our molecular tumor board locally as well as the monthly R cancer genomes. And we discuss many complex cases, looking at both, again, germline and tumor next-gen sequencing reports. We have access to, again, a multitude of different expertise, including molecular genetics, clinical genetics, bioinformatics, et cetera. And this also struck me that we had new opportunities to help cancer patients. And the first example of this, shortly after I arrived here at Vanderbilt, liraglutide, that drug that you just saw in the case report, became approved in this country. And so because all of these data are stored electronically in our center, and we have such amazing bioinformatics people here, we went back and we actually could find additional cases where NTRK was translocated. And we notify the providers and we tell them that their patients are potentially candidates for these drugs. And there are even patients that you might not think, like cancers, more common breast cancers, that can have these fusions and rearrangements. This also presents new opportunities for breast cancer and immunotherapy. I think despite the fact that we do have one indication of triple-negative breast cancer in the metastatic setting, I think most of us know who have treated triple-negative breast cancers with immunotherapy know that it is not a huge drug in terms of people having the great majority of patients having dramatic responses. There are a couple, but not many. And so getting better molecular predictors of who's going to respond to immunotherapy is incredibly important. Why that is they're expensive, they can have significant side effects, including sometimes death, as I think anyone in the audience who have treated enough of these patients has seen. Sequencing of these tumors may in fact allow us to better select patients for immunotherapy, including in breast cancer. And so a seminal paper came out of the New England Journal of Medicine from colleagues at Johns Hopkins, notably Bruce Augustine and Luis Diaz, as well as Young Lee, the first author. They were really the first to put on this concept that tumor mutation burden may predict for response to immunotherapies. This actually was a very elegant and simplistic hypothesis because it's kind of like playing the lottery is how I like to think about it. The more tickets one buys, the higher the chance that any one of those lottery tickets will actually be the winner. And so when we think about tumor mutation burden, what that is really measuring is how many DNA mistakes are in that individual's cancer. The higher the number of DNA mistakes, the more likely that any one of those gene mistakes will lead to a neoantigen or something that the T cell can recognize that's on the tumor. And so the therapies that we give for immunotherapies block a specific interaction that tells tumors that this is, has the tumor tell the T cell, don't attack me because I'm you. And so if we block that interaction, and there's probably a dozen of these so-called handshakes, as we call them, we can actually induce autoimmunity. Now, again, we have to be careful because autoimmunity is a side effect of this and can sometimes even be lethal. So using one of a dozen handshakes can sometimes give us enough immune response against the tumor and not one cell. But again, that really relies upon having a neoantigen. So the rationale, the higher number of DNA mistakes, the higher the likelihood that any one of those will in fact elicit an immune response because they are an effective neoantigen. And so that was also put in the test for just breast cancer, breast and other cancers. In this particular trial, these were all patients who had mismatch repair deficiency or microsatellite instability, as it's often called. That is a hallmark of DNA repair deficiency genes leading to increased tumor mutation burden. There are, in fact, other ways to get high tumor mutation burden. So subsequent trials have looked at this, including in patients who had breast cancer. This was a patient of mine. and this is her Foundation Medicine report. And I think I just want to highlight that she had, even though she did not have deficiencies in DNA repair mechanisms, as noted here by the microsatellite stability status, she did have a fairly high, at the time, the highest that I had seen, but now that I've seen much higher tumor mutation burden at 68. And so this was her PET scan. Before she started immunotherapy, she had this large, very symptomatic mass in her psoas muscle. And you can see by May 2018, it had complete resolution. And she had about almost 2 years of disease-free progression, or progression-free survival, I should say. And she did great. She unfortunately did eventually progress. This was something that we had not seen before in breast cancers, especially in estrogen-positive disease. And going with the same paradigm that I showed you about how these therapies become FDA approved, that we have all electronic data on all our patients, we can go back. And so we did the same thing when the MSI approval came by the FDA for these therapies. And more recently, just this year in this country, one can now have MSI stable or MSS, but still have a high tumor mutation burden and get these immunotherapies in the country. So just highlighting again that breast cancer can fall into these categories and have real meaningful clinical responses to immunotherapies based upon a high tumor mutation burden. The other thing I want to highlight is that we need to start sequencing more. We know that there are some centers, there are some providers and patients that know about next-gen sequencing, but across this country, and probably I would argue across the world, this is still relatively uncommon. And why is that important? This is a paper that we actually published looking at just patients with metastatic breast cancer and not falling under what we currently call guidelines. This is at the time to see how many patients would in fact have germline alterations. And we did find more patients with germline alterations than one would have found if one just followed our or then current NCCN guidelines. Subsequently, our guidelines have changed, that now every metastatic breast cancer patient is recommended, if it's going to alter therapeutic modalities, to undergo genetic testing. The interesting thing is, even though none of our patients who did not meet in surgical way, all of our patients who had a BRCA1 or 2 mutation did in fact meet NCCN guidelines for germline testing, but there were a couple BRCA2 variants that in fact did not get tested. So this is an additional problem in this country, and again, arguably the world, of dissemination and implementation. Why are these patients not getting tested when they meet categorical guidelines and should? And now that we have PARP inhibitors, this could actually have dramatic effects for their therapy and longevity with metastatic disease. So this is a real problem, and one that we are tackling here at Vanderbilt and across the country and the world to try to what I would use the term democratize genetic testing for cancer patients. And so that's really the vision of precision oncology that I have at our cancer center, is that using the internet, telemedicine, and also genetics for the genetic side, get all cancer patients into our clinics and sequence, get the tumor testing using our robust data science applications and bioinformatics. We have 2 forms of organ information flow, and then convene our molecular tumor boards, both our local and our worldwide, and then recommend a bunch of different modalities, innovative and pragmatic clinical trials, targeted immune therapies, as well as risk management on the germline side. Also thinking about how do we actually get more patients, and we're working with the concept of, can we actually go direct to patients to make sure that they know that there are options for their metastatic cancer and treatment modalities. And so the future of GCI is here. Last year, myself and my colleague, Dr. Dizon, who I see is on the call now, were asked to become the new chief medical officers for Paul Goss, who is stepping out from the roles. I'm leading more of the breast side, which many of you on this audience know because I've attended your tumor boards as well. And then Tom is doing the gynecologic program. So in conclusion, hopefully I've convinced you that tumor testing and germline testing can in fact yield actionable variants, both from a screening standpoint as well as a therapeutic modality standpoint. However, great care really needs to be used when interpreting these tests. I firmly believe that the establishment of molecular tumor boards can aid in this decision-making process, providing the needed content expertise. And then I also really am very, very excited about the use of telemedicine and how we can actually get testing tumor and germline for all patients across the world. So with that, these are my acknowledgments. There's just a ton of people to thank, both in my lab, other labs, clinical and laboratory collaborators listed here, but also patients and, of course, local different funding sources.